Hi everybody, and welcome back for our discussion of Chapter 2, Fuel for Exercise, Bioenergetics, and Muscle Metabolism. To start our conversation, we need to discuss the substrates that we utilize for fuel for physical activity and exercise. Our three main substrates are carbohydrates, fats, and proteins. These three substrates are made up of carbon, hydrogen, oxygen, and nitrogen, which are going to contain essential bonds that hold energy in our stored food. When we release these chemical bonds from our food, they will produce high-energy compounds such as ATP. When we are at rest, our preferred substrates are about 50% carbohydrates and 50% fats. However, this starts to change with participation in physical activity and exercise. When we go through short, high-intensity bouts of physical activity or exercise, we tend to rely more on carbohydrates. As we go towards long, slow, endurance-type activities, we tend to utilize a combination of carbohydrates and fats. The longer the duration of the exercise or the lower the intensity, the more we tend to prefer using fats as our preferred substrate. Looking more closely, carbohydrates, when we consume them in our body, will all be converted to glucose in the form of blood sugar. This is going to give us about 4 kilocalories per gram of energy. We can also store glucose in our body as glycogen. This is going to contain about 2,500 kilocalories that we are able to store in our body. Carbohydrates are the primary ATP substrate preferred by the muscles and the brain. When we consume extra glucose, as I said, it will be stored as glycogen, and this tends to hang out in the liver and the muscles. Glycogen can then be converted back at any time to glucose when we need it to make more ATP. Our glycogen stores are limited to about 2,500 kilocalories, so any dietary carbohydrates need to be consumed in order to replenish glycogen stores. Any excess carbohydrates consumed past what the body can store as glycogen will be converted into fat. Fats, on the other hand, are our most efficient substrate and have the most efficient form of storage. These give us about 9 kilocalories per gram of energy, and we can store up to 70,000 kilocalories or more in our body in the form of adipose tissue. The energy substrate for fats gives us prolonged, less intense activities. It has a very high net yield of ATP, but the production of its ATP is very slow. We also have to take fats and break them down into free fatty acids and glycerol. Only our free fatty acids can be used to make ATP. Finally, proteins are our energy substrate that are used during starvation, so we don't often use proteins for energy. However, when we do, this will give us about 4 kilocalories per gram of energy. We have to take proteins and convert them into glucose through a complex process called gluconeogenesis. Additionally, we can convert free fatty acids from sugar for energy storage. We also can use this for energy substrates. This table will help give you an idea of where energy is stored in the body in the form of either glycogen or fats. So, if I could stress a most important table for you to study for this entire class, this table would be it. I would pause here and copy this down. This is incredibly important. So this table is going to demonstrate the entire process of going from the food we take, breaking it down into our three major substrates, what their building blocks are, and then how we get this to actual ATP and metabolism. So we know that our metabolism is the summation of all of the chemical reactions that occur in our body. We need our energy from these chemical reactions to be released at a controlled rate, and this is going to change based on the availability of our primary substrates. This is going to work through the law of mass action effect. Basically, substrate availability is going to affect our metabolic rate. So the more available substrate I have, the higher that pathway of activity. So for example, if I consume a Snickers bar, if you think about Snickers, what is there going to be a high amount of in Snickers bars? Well, there's maybe a little bit of protein from the peanuts. There's maybe a little bit of fats from like the cocoa butter, but there's going to be a whole lot of sugar, which is a carbohydrate. So when you think about it, which pathway is my body going to take to make energy? It's probably going to be the carbohydrate pathway. So this is an example of mass effect in action. So if we have any excess of a given substrate, the cells are going to start relying on that substrate more than on our other sources. So if I frequently only consume carbohydrates or very high amounts of carbohydrates, my body is going to come to depend on that fuel source. Another important concept in this discussion is enzymes. So enzymes are basically biological catalysts. They allow energy to be released at a controlled rate based on what metabolic pathway we're using. So enzymes don't actually create energy, they just make it easier for a chemical reaction to happen. They do this by lowering activation energy. So they do not start a chemical reaction or set an ATP yield. They just facilitate the breakdown of a particular substrate. So, and again, this is by lowering the activation energy for that particular chemical reaction. We can typically identify any enzymes when we're looking throughout anatomy and exercise physiology because they will end with the suffix ACE, A-S-E. For example, ATP is broken down by the enzyme ATPase. There will be a specific enzyme or chain of enzymes required for each step in any biochemical pathway. So the more enzymes in that particular activity, the more products that will be produced. There are also specialized enzymes called rate-limiting enzymes. These are going to create a bottleneck effect on any step in the biochemical pathways. Their activity is going to be influenced through something called negative feedback. This has the ability of slowing the overall reaction and prevents any runaway reactions in the body. So, this is an example of a typical metabolic pathway, and this is going to show the important roles of enzymes in controlling the rate of reaction. In this example, you see an input of energy in the form of stored adenosine triphosphate, or ATP, and it's going to be needed to begin a series of reactions. This is your activation energy. But this will be less initial energy without than if we had one or more enzymes involved. As fuels are subsequently degraded into byproducts through this metabolic pathway, so the enzyme breaks down the fuel, you're going to see ATP formed. The utilization of any stored ATP is going to result in the release of usable energy, heat, and a release of an adenosine diphosphate, or ADP, as well as an inorganic phosphate. This process will continue, taking the fuel to byproduct until eventually you reach a rate-limiting product. This is eventually going to come back and impact the enzymes at the beginning of the reaction. This is a negative feedback loop. Looking forward, what is actually ATP? Our body only has the ability to store very small amounts of ATP until it is needed. When we break down ATP, it is to release energy. ATP plus water plus ATPase is eventually going to produce ADP, an inorganic phosphate group, and energy. ADP is the lower energy compound and is much less useful for what we use for our biochemical reactions. If we want to synthesize ATP from its byproducts, we just flip the reaction the other direction. ADP plus an inorganic phosphate group plus energy will give us ATP, and this process is called phosphorylation. This can occur either in the absence or presence of oxygen. We would call this an anaerobic or an aerobic reaction. So, we have a few different energy systems of the body. We know the body has limited ATP storage, so we constantly have to synthesize new ATP. These three ATP synthesis pathways are the ATP-PCR system, which is an anaerobic metabolism system, the glycolytic system, also known as glycolysis, which is another anaerobic metabolism system, and the oxidative system, also known as aerobic metabolism or cellular respiration. Looking at these more specifically, the ATP-PCR system is anaerobic substrate level metabolism. For every one ATP, there is going to be one PCR. This ATP-PCR system only works for about 3 to 15 seconds, and this is because the ATP stores are very limited in the body, so this pathway is used to reassemble ATP quickly. This is going to use a PCR, also known as phosphocreatine. Phosphocreatine plus creatine kinase is going to make a creatine plus an inorganic phosphate plus energy. The PCR energy cannot be used for cellular work, although it does allow us to reassemble ATP quickly. This will give us the ability to replenish ATP stores very quickly during rest. This also allows us to recycle ATP quickly until it is used up, again, somewhere between 3 and 15 seconds. Looking at this system, we know it's an anaerobic substrate level metabolism. This is because oxygen is not needed to make this process occur. This process is going to occur in the cytoplasm of the cell. So this does not occur anywhere special. This is happening frequently within the cell. The other important concept of PCR is it's going to be catalyzed by creatine kinase. Creatine kinase is going to control the rate of ATP production. This is our rate-limiting enzyme. This is going to create a negative feedback system. So when ATP levels start to increase, or ADP levels start to drop, the creatine kinase activity will also drop. The inverse will happen as well. Our second system is our glycolytic system or glycolysis. This is our other anaerobic system. Its ATP yield is 2 to 3 ATP for every glucose. This works somewhere between 15 seconds to 2 minutes of physical activity. This is going to break down glucose via the process of glycolysis. We can use glucose or glycogen here as our substrate. However, starting with glucose or glycogen, it has to go through a conversion step into glucose 6-phosphate. This conversion from glucose or glycogen to glucose 6-phosphate is going to cost 1 ATP. Glycogen, however, doesn't have a cost, so that will cost us 0 ATP. This is why there's the difference between a net of 2 or 3 ATP. Going through the process, the pathway will start with the glucose 6-phosphate and end with a pyruvic acid. There are somewhere between 10 and 12 enzymatic reactions that go through the process of glycolysis. For the purposes of this class, you do not need to know all 10 to 12 enzymatic reactions. You just need to know that we go from a glucose 6-phosphate to a pyruvic acid. All of these steps are also going to occur in the cytoplasm. The ATP yield again is 2 ATP for glucose and 3 ATP for glycogen. The cons of using the glycolytic system is that we have a very low ATP yield, so we're using our glucose inefficiently. Also, the lack of oxygen converts our pyruvic acid to lactic acid. This lactic acid can impair glycolysis and ultimately muscle contraction. So if we use the system too much without the presence of oxygen, the system actually shuts itself off. However, the pros of the glycolytic or the glycolysis system is it allows our muscles to contract even when oxygen is limited. It also permits shorter-term, high-intensity exercise than our oxidative metabolism would normally sustain. The most important rate-limiting enzyme in the glycolytic system is phosphofructokinase, or PFK. This is going to change, so when we have high levels of ATP, PFK activity will decrease. When we have low levels of ATP, PFK activity will increase. This will also regulate the products of the Krebs cycle. Finally, our third system is our oxidative system, also known as our aerobic respiration or cellular respiration system. The ATP yield is dependent on the substrate in this particular system. If we're working with glucose, we're going to get somewhere between 32 and 33 ATP. If we're utilizing something like a free fatty acid, we could end up with hundreds of ATP. Our oxidative system allows for a steady supply of ATP for hours and sometimes even days at a time. It is the most complex of our 3 bioenergetic systems. This system is going to occur in the mitochondria, not exclusively in the cytoplasm. Within the mitochondria, we're going to rely on the density within the mitochondria to determine the location and the oxygen diffusion. We also want to minimize excess oxygen to prevent creating reactive oxygen species within the mitochondria. So this is why the mitochondria have all the lovely little folds in it. It allows us to have high levels of interaction without having too many reactive oxygen species. So the first stage of our oxidative system is actually going to be glycolysis. So we would go through the glycolytic system like we would with anaerobic respiration, but this time oxygen is present. This will allow us to go to stage 2, which is the Krebs cycle, and then ultimately stage 3, which is the electron transport chain. So as we've stated before, glycolysis can occur either with or without the presence of oxygen. The ATP yield will be the same for anaerobic glycolysis as well as glycolysis in the presence of oxygen. Also, the steps will be the same as anaerobic glycolysis. But if oxygen is present, pyruvic acid will be converted into acetyl-CoA, and that acetyl-CoA will enter the Krebs cycle. In the Krebs cycle, one molecule of glucose will produce 2 acetyl-CoAs. This means that one glucose molecule will turn the Krebs cycle twice. This also means that one molecule of glucose will double the ATP yield typically reflected in the Krebs cycle. This means 2 acetyl-CoAs will produce 2 GTPs and 2 ATPs. Also produced during the Krebs cycle is NADH, FADH, and hydrogen ions. If we have too many hydrogen ions in the cell becomes too acidic. Therefore, we have to move these hydrogen ions to the electron transport chain, which is the last step of aerobic respiration. Looking at the electron transport chain, these hydrogen ions produced in the Krebs cycle are transported via NADH, FADH2, and other carrier molecules. Hydrogen ions then travel down the chain, and this will combine with oxygen to neutralize and form H2O or water. The electrons plus the oxygen will help to form ATP. There are also about 2.5 ATP per NADH and 1.5 ATP formed per FADH2. This means through the entire electron transport chain, we are going to produce somewhere between 30 to 34 ATP. Looking at aerobic respiration summary, this is a good slide that kind of summarizes all of the process. One glucose will give us around 32 ATP, one glycogen will give us around 33 ATP, and then we will have a couple net totals. Glycolysis will give us 2 or 3 ATP. GTP from the Krebs cycle will give us 2 ATP. 10 NADH will give us 25 ATP. 2 FADH2s will give us 3 ATP. And this is how we come to our total summary. This is another slide that kind of breaks down where everything is coming from. Again, this is the time where I would pause the video and take some notes. Finally, we don't only use glucose, if you remember, our carbohydrates to get our energy during oxidative respiration. We can also utilize fats and proteins. For fats, we tend to use triglycerides as our major fat energy source. We break down our triglycerides into a glycerol and 3 free fatty acids. Lipolysis, or the breakdown of fats, can only be carried out by particular enzymes called lipases. The rate of free fatty acid entry into the muscle will be dependent on the concentration gradients. Also, we know that fats are going to yield somewhere between 3 to 4 times more ATP than glucose. However, utilizing fats is much slower than glucose oxidation. This is because we have to go through the process converting free fatty acids to acetyl-CoA before it can enter the Krebs cycle. This means we have to have an upfront expenditure of 2 ATP. The number of steps dependent to create acetyl-CoA from our free fatty acids depends on the number of carbons in our free fatty acid. For example, a 16-carbon free fatty acid will yield 8 acetyl-CoA. In comparison, 1 glucose only yields 2 acetyl-CoA. That oxidation is also going to require much more oxygen, so it will yield more ATP later. This is why when we have long, slow duration exercise, fats tend to be the preferred energy substrate. Our acetyl-CoA from our fat will enter the Krebs cycle. Then it will follow the same pathways as glucose oxidation. Different free fatty acids have different numbers of carbons. This means the yield will be different numbers of acetyl-CoA for every different free fatty acid molecule. So, for example, our palmitic acid has 16 carbons, so its net yield will be somewhere close to 106 ATP. Finally, we can oxidize proteins to be used as an energy source. However, we very rarely use this substrate and only use it in a state of starvation or extreme energy expense. We have to do this by taking the protein and converting it into glucose via gluconeogenesis, and then this glucose is converted further into acetyl-CoA. We can't really determine an energy yield for protein. We know the presence of nitrogen makes this process very unique, and then we have to take the nitrogen and excrete it out through our kidneys, which is going to require ATP expenditure. So because it is generally minimal, estimates typically ignore protein metabolism when we talk about energy substrate use. Finally, that lactate that is produced during glycolysis, specifically anaerobic glycolysis, can also be utilized as an important fuel source during exercise. Our muscles can turn around and use lactate in 3 different ways. Any lactate produced in the cytoplasm can be taken up by the mitochondria of the same muscle fiber and then oxidized. The lactate could also be transported via an MCP transporter to another cell and oxidized there. This is a form of a lactate shuttle. So basically the lactate's not being well used in this one cell, so I ship it out to another one. Lactate could also be recirculated back to the liver, so I take it out of the cell and put it into the bloodstream, and then I reconvert that into a pyruvate, and then we turn the pyruvate back into glucose through gluconeogenesis. Our negative feedback is going to regulate the KREBS cycle and how our lactate is used. Isocitrate dehydrogenase is our rate-limiting enzyme when it comes to lactate utilization. This functions very similar to PFK for glycolysis. This will also regulate our electron transport chain. Lactate utilization is inhibited by ATP, but activated by ADP. So lactate tends to be a preferred energy source when we are struggling to supply energy in other ways. Finally, I want you to think of our energy systems as kind of a continuum. All 3 of our energy systems are going to interact. No single system is going to contribute 100% of the energy produced at any given time. However, during certain types of activity, one system tends to be more dominant. This is because the cooperation is going to increase during transition periods. So for example, if I wanted you to think about if I were to jump up out of a chair, which system would you use? Take a moment. Hopefully you said your creatine kinase system or your PCR system, right? If I jump up suddenly out of a chair, that's less than 2 to 15 seconds, even though I still have some energy coming from my aerobic metabolism system to make sure I'm digesting my lunch or whatever else is going on in my body. I utilize the PCR system to jump up out of the chair. Now, let's say I am now a mile runner. If you start to think about that, that's sitting in a weird spot somewhere between glycolysis and our aerobic respiration systems. All right, going through that process, if I were to kick at the end of the race, I'm starting to lean into my PCR system a little bit. So really take it and think about this as a continuum going forward. All right, so this is a nice summary that kind of summarized that this is a transition from side to side. Also, is oxygen present for this particular process? Is it not? So take this time to kind of look over this slide. It's another good study slide. Finally, an important concept to note is the crossover concept. So basically, there is a point with physical activity where we go from preferring carbohydrates as our energy source or fats as our energy source. So at rest and exercise below 60% of our VO2 max, lipids tend to be our primary substrate. That is our preferred energy source. However, as we start to move towards high-intensity activities, moving above 75% of our VO2 max, carbohydrates start to serve as our primary energy substrate. This crossover point is the intersection where we are affected by exercise intensity and endurance training. This is where we go from a fat preference to a carbohydrate preference. And finally, all of our muscles have a different capacity for oxidation. Not all muscles exhibit maximal oxidative capacities. This is because our enzyme activity is going to predict our oxidative potential. We have certain representative enzymes like succinate dehydrogenase and citrate synthase that are going to kind of be our predictors here. Our levels will be different in endurance trained versus untrained individuals. When we have lots of type 1 fibers, we tend to have greater oxidative capacity. There also tends to be more mitochondria and higher oxidative enzyme concentrations. Type 2 muscle fibers, on the other hand, are much better at glycolytic energy production. So what type of muscle fibers that you tend to have will also predict what type of energy system you prefer to use. If we were to take somebody through endurance training, we know that this can enhance oxidative capacity of our type 2 muscle fibers. So remember, we can train type 2 muscle fibers to be better at endurance. This will also allow them to develop more and larger mitochondria. This will also develop more oxidative enzymes per particular mitochondrion. Right? ATP demand will increase with intensity. So in response, increases will occur in the rate of our oxidative ATP production, our O2 intake of the lungs, and our O2 delivery by the heart and the blood vessels. We know that O2 storage is limited in the body, so I either use it or I lose it. So oxygen levels entering and leaving the lungs are going to enable accurate estimate of O2 use in muscle. Therefore, this is why we do things like use the metabolic cart during aerobic exercise bouts to estimate somebody's substrate use. So, thank you everybody for your attention, and I look forward to hearing back from you guys for Chapter 3.